Here we go, my friends. We're rolling through week number 2 and chapter number 22. We did a little overview of the flow of lymph and how lymph is produced, what lymph is, the lymphatic system with immune function, and now we're really going to talk about how we talk the organs and the tissues, the structures of the lymphatic system. We've talked about the lymphatic capillaries and the vessels and the trunks and the ducts, but let's talk about the more traditional structures or organs or tissues that are involved in the lymphatic system and that really are the sites of immune function. And it's these 5, and they break out pretty easily. And we'll just take these 5 on the left, 5 organs or tissues, and we will discuss them as either secondary or primary or secondary lymphatic organ, or just general tissue if it doesn't quite reach organ stage. And then we'll take those same 5 on the left, and we'll talk about characteristics, and we'll decide which of those 5 have no afferent lymphatic vessels, which of those 5 don't have surrounding surrounding connective tissue capsules, which of those 5 are the sites of maturation of T cells, and so on as we go through. That's how we'll test ourselves. And the way it breaks down, there are 5 structures that were given to you. 2 of them, the red bone marrow and the thymus, are primary lymphatic organs. 2 of them, the lymph nodes and the spleen, are secondary lymphatic organs, and then 1 of them, lymph nodules, is a very general non-organ term. It just means tissue within the lymphatic system gathered together, and they're called lymph nodules, and you find them throughout the body. How do we break and you can see they're all going to be split out here on this image. So the bone marrow, I'll go ahead and circle the bone marrow, and again the epiphyses of the long bones and in the flat bones. And the thymus is going to sit right here. I'll blow this up in just a moment. I'll circle the thymus. Those are the 2 primary. And then the spleen, and then all these little dots, all these little beans. We'll see these bean-shaped little dots that you see along the lymphatic system. And I'll show you those in a little detail. Those are all, so the spleen and those lymph nodes are the secondary lymphatic organs. And then there's lymph tissue spread throughout the whole mucosa of your body. So the 2 primary are the red bone marrow and the thymus. And why are they primary? Well, that's because that's where cells, B cells and T cells, immune function is all about B cells and T cells. B cells and T cells. And essentially, B cells and T cells get together to make antibodies, and that's called humoral immunity. And then T cells become cytotoxic T cells, and they kill cells phagocytotically, and that's cell-mediated immunity. So this is roughly how it breaks down. We'll talk about that quite a bit more. But the red marrow makes both the B and the T. It matures the B in the bone marrow, and then it makes pre-T that are ready to go, but not quite ready to go. They're half ready to go. And then the bone marrow sends those T cells. The B cells are ready to go. So the bone marrow sends out the B cells fully armed and ready to go. But then the bone marrow sends the T cells out to the thymus to be matured at the thymus. So if you want some tips on where they're matured, bone marrow matures the B cells, and the thymus matures the T cells, B and T, if that works for you. And from the thymus, once the T cells are matured, then they move out, they develop into what's called immunocompetent cells, meaning they're ready to go. And then at that point, the B from the bone marrow and the T from the thymus are then sent to the secondary lymphatic organs, the lymph nodes and the spleen, to post up and dig in and go into those organs and become functional immune tissue, right? Part of the lymphatic system, and they're going to start targeting foreign antigens or foreign microorganisms, foreign bodies. And the bone marrow and the thymus are going to be those 2 primary, and the bone marrow, of course, blown up here. You're going to see the epiphyses of the long bone showing the femur here. And then the thymus is this 2-lobed, butterfly-shaped green organ. If you see that green, it looks a lot like the thyroid gland, but a little bit bigger. And I'll show it to you in more detail in the next slide. But there's this organ right there, and it's bilobed, and it's like a butterfly, and it sits right above the heart, and that's the thymus. That's where the T cells are maturing. Okay. And that maturation process is exactly what we saw way back in 40B. Not way back, but back in 40B, where you had the pluripotent stem cell. Forget about the whole left-hand side. Forget about your myeloid stem cell and your granular. Come over here to the agranular, the non-granular cells, and recall back to the lymphoid stem cell line. And you have your natural natural kill that we'll talk about in a little bit. But really 2 of the 3 you're producing are your T's and your B's, and they're both produced in the bone marrow. The B's are polished off in the bone marrow and matured. The T's are sent to the thymus for maturation. And that's about as simple as it gets. And what we'll do is move. So we'll take a look at the thymus. The bone marrow we've seen in detail, and it's really just that schematic that you saw of producing the B and the T cells and developing them from the lymphoid cell line. The thymus is going to be this organ, and here it is shown. It's green on the that lymphatic system image that I just showed you. Everything is green, which is kind of weird to see the bone marrow green. Weird to see the spleen green. They're not green. They're just a symbolic way. The thymus is going to be shown, again, right in this position. It's going to sit within the mediastinum. It's going to be completely posterior to the sternum. It's going to be superior to the heart, medial to both lungs. And it's going to be sitting just anterior to the branching of the trachea. So the trachea will be shown. I'll trace where the trachea would go, would come down, and of course it would branch. The trachea would branch into the 2 main bronchi, and right, literally sitting directly anterior to that branching would be this thymus. Okay? And this is where the immature T cells, the pre-T cells, are going to come from the bone marrow, and this is where they're going to mature. And how is it broken out? You see the 2 lobes very clearly there. They're separated by a connective tissue capsule, and the connective tissue capsule wraps over the thymus superficially. But, as we've seen before, and we'll see quite a bit more, it penetrates down into the thymus. So that connective tissue penetrates down into the core of the thymus, and it becomes trabeculae. And that divides the lobe, each of the 2 lobes, into lobules. So, for instance, this lobe here would be divided into these various lobules. I'm drawing all these lobules or lobules here that is going to be divisions of the thymus. And that's going to be created by the connective tissue capsule penetrating down into what's called trabeculae. And that's going to be seen in this image on the left. The connective tissue capsule, likely dense irregular, is what it tends to be when you have a capsule around an organ, is going to be running along the superficial surface, and then you're going to dive straight down into the heart of the matter, into the thymus. That's a trabecula there, and here's another trabecula running through, and you can see it's dividing this particular lobule. So this is a lobule here, divided up, and I don't know where it goes on the left, but it's divided up by the trabecula. And within each lobule, I'll go with lobule from now on, you're going to have such a traditional anatomical breakdown. A superficial cortex around a central medulla. We've seen this before. We'll see it quite a few times in this slide, in this lecture module. You'll see it a few more times, and we'll continue to see it. You're going to have the superficial cortex surrounding the core medulla, and the medulla shows up light in this image, lightish pink, and the cortex shows up darker purplish or so. And there are going to be 4 types of cells running around the thymus. So you're going to want to have a rough idea of these. They break down super easy. It's the T cells that are all put there from so the thymus is just a collection of T cells that came from the bone marrow to mature. And so you just have infinite numbers seemingly of, not really, but seemingly infinite numbers of T cells filling the thymus and all waiting to mature, all waiting to be matured inside this organ. And the 2 cells that are doing the maturation, that are facilitating that maturation, are the dendritic and the epithelial cells. You see on the right inside the medulla, an epithelial cell is going to be shown, large epithelial cell. And what are those 2 doing? We'll get into this in more detail when we talk about development of T cells or maturation of T cells. But really they're going to be tested. These T cells are going to be tested on 2 key questions. 1, can you recognize an antigen? Can you recognize and identify a foreign antigen, a foreign body that may kill us? Can you recognize it, and can you go to work battling it? And then if you pass that test, you go to the next room and you get a quick little test that says, oh, hey, check this out. Do you want to kill this? And what it is a self molecule. And if you get all wired up and you try to kill that, you get booted out, because you need to be able to decide, this is a foreign antigen, I'm going to kill this, but this is myself and I'm going to leave it alone. And it's only the T cells that can make that split. They can go after foreign antigen and kill a foreign antigen, but they can pull back and go, whoa, that's me, I'm not going after that at all, because if you let those T cells through that are going to destroy self molecules, you're just opening the door wide open to the incredibly vast array of autoimmune diseases that you really don't want to be near any of them if you can avoid it. Right? So 98% are going to fail because it's a tough test, and 2% are going to pass. So 2% of T cells are going to pass this test and become immunocompetent or functional. You're going to sort of collect to some extent in the thymic corpuscle, which is this structure that I'm outlining, large aggregation of T cells, and from there it's sort of a central hub, and they'll be sent out to go post up and root inside of the secondary lymphatic organs, the spleen and the lymph nodes, and that's where the T cells will go to work. What happens to the 2%, the 98% that fail? Well, they're going to be broken down. Apoptosis is going to be initiated. Apoptosis will become very important in this module, this week's modules. It's programmed or ritual or purposeful cell suicide or cell death. It's the cell undergoing a set of steps that kills itself on purpose. And that's what the 98% of failed T cells do that cannot become immunocompetent. And so they break down, and now your macrophages are the fourth cell type inside of the thymus, and they go to work just cleaning up all that cellular debris of the 98% of the cells that fail the test. And again, the thymic corpuscle kind of gathers together the 2% that pass, and they send them out to the lymph nodes in the spleen, the secondary organs, where they colonize those organs, and they set up shop, and they get ready to just sit there as guard duty and just start attacking everything that comes through your body and tries to kill you. And then one quick note about the thymus before we move away from the thymus, and this is completely not really related to this is very tangential, but you should know that the thymus ontogenetically ontogenetic means within the course of a lifetime. Ontogenetically, the thymus changes drastically. It is an incredibly front-loaded organ towards the beginning of your life. It does the vast majority of its work before you're even 14 or 15 years old. And then it just goes into cruise mode and kind of fades to black. By the time the thymus matures, tests all those T cells from the bone marrow, matures the 2%, fails the 98%, and then sends out the good 2%, that's all happening almost prepubescently, or even maybe as you're entering puberty, but not too much as a post-pubescent person. And that's all being done early on, and then the thymus just starts to degrade. It's essentially that the parenchyma of the thymus, the tissue that's going to work, is slowly being colonized by adipose tissue and loose areolar connective tissue, and the organ just sort of becomes defunct over time, and the older you get, the less the organ actually does. So, anyway, just something to think about. And lymph nodes. So now we're on to the secondary lymphatic organs. Now we've said, okay, we finished the B cells because we got that done in the bone marrow. We finished the T cells. We got that done in the thymus. Now we're sending them on out. Where do we send them to? Spleen and lymph nodes. And let's talk about the lymph nodes first. What are they? They're these 600 or so tiny little bean-shaped little swellings right along the lymphatic vessels. It's just a picture a road going and having a little swell out to pull off to the side on the road. That's all. That's all it is. It's just a lymph vessel swelling out into a lymph node. And you can see 600 of them. This image is not going to show 600. I'll circle maybe, I don't know, I'll circle 5 or 6 or 7 or 8. You see them throughout the body, these little swellings, and where they aggregate, or where they congregated, or where they tend to show up more often. You should probably maybe know this just from general pediatrician visits when you're sick. You know, under the lymph nodes under the jaw, and in the armpit, and in the groin. Those are typically, and around the mammary glands to some extent too in females. But really, under the jaw, armpits, and groin, that's where you check the main areas for swollen lymph nodes. And even though swollen lymph nodes from, again, way back when you were a child going through pediatrician visits, swollen, looking for swollen lymph nodes, and your physician giving you the check there and checking to see if these are swollen. 600 or so smallish bean-shaped organs plumbed right in line with the lymphatic vessels, which is nice, and concentrated heavily in the areas that I just dictated or just mentioned. And it just tells you that every single milliliter of lymph in your body is constantly filtered through these things, constantly filtered through. And what does that filtration process look like in terms of flow? Well, afferent vessels bringing in, and I think that showing 5 here, 1, 2, these are cut open so you can see the one-way valves. 3, 4, 5 afferent vessels bring the lymph in, and they cruises through the subcapsular sinus, into the trabecular sinus, into sort of the medullary sinus in the medulla, and then out through. So basically the flow is through. I'll just put some arrows here. The flow is through. This would be in a roughly left to right direction. The flow is through all this tissue interacting with the cortex and medulla. I'll talk about in a second. And all these coalescing through and leaving through these two efferent lymphatic vessels that continue the movement of the lymph on through the progression that we saw last module, right? On through the trunks and then into the ducts and back into the blood and so on and so forth. So you have the outer tissue connective tissue capsule, and that's clearly shown here in this image. In green is going to be the outer tissue, and then that's going to surround, and then you have same thing, superficial cortex and a deeper medulla, and that's going to show up in this image where the cortex is going to be roughly this structure here is going to be roughly the cortex. I'll make it like a donut shape because that's kind of what it is. The donut shape of the cortex would fill in all this area here like a donut. Roughly that's the cortex. Leaving the center is all medulla in the middle. So cortex wrapped around medulla in the middle. Same story we've heard over and over again. The cortex and the medulla are going to be the parenchyma of the organ. If you don't remember that, go all the way back to 40A. The parenchyma is the working functional tissue. And I'll jump to the bottom. That's connective tissue capsule, the trabeculae, and the reticular fibers, and the fibroblasts are going to be the stroma. That's the supporting tissue, the structural supporting tissue, right? Parenchyma versus stroma. Again, if you're unsure, go all the way back to 48 to review that. And then, we've seen this before too. This is some points that just should be familiar. Here's the connective tissue capsule. Oh, look, it's diving down into the core of the organ here, the lymph node, as trabeculae. Trabecula, single, trabeculae, plural. And in it goes, diving down and in, and it's coming down and making these divisions and dividing the lymph node up into various sections. Right? And the flow again, the lymph enters the node, flows through the subcapsular sinus. So you're coming in, you're flowing into the subcapsular sinus about here. Then you're flowing down in through the trabecular sinus. And then you're eventually going to flow even further. But as you flow through the trabecular sinus, you're exposing yourself to the outer cortex. And so this is the outer cortex. Or this area here would be the outer cortex. So as you flow down through, I'll put an arrow there as you're flowing through. As you're flowing down, hopefully that arrowhead is showing clearly that I'm trying to indicate that you're flowing downward into the lymph node. As you flow down, you're going right by the outer cortex. And what is posted up in the outer cortex is going to be a bunch of B cells. And those are going to be incredibly important for recognizing the antigen, and starting up that whole cascade of adaptive immunity that we'll talk about in a few modules, whereby you're going to create the correct antibodies against the antigens that are coming in and trying to invade and take over your body. So as the lymph flows down through into through the outer cortex, through the trabecular sinus, it will run through the outer cortex and will run into all these B cells that are all posted up and in this reticular mesh. Picture a reticular mesh like a colander, and all these B cells just sticking right there, and the lymph has to flow over the B cells, and the B cells get a taste of every bit of that lymph, and they can recognize, hopefully, every antigen that comes through. Okay, and that can be seen in a nice view here of a micrograph. Again, in blue is the capsule, and here's the trabecular sinus. The lymph would flow through the trabecular sinus. Over here, let's say the capsule ran across. Trabecular sinus would run like that, and the lymph would flow through. All the way in sort of reddish purple up here, they're all the that's all the outer cortex. And there's something called a germinal center, and this is called lymphoid or lymphatic nodules make up this outer cortex, and they're just B cells posted in there and stuck in there constantly to filter that lymph. Okay? As you flow even further down the trabeculae, you start to enter this purple region. Right? So this purple region, I'll kind of outline it right around in here. This is all the inner cortex, the beginning, kind of the transition between this reddish purple and the actual purple. That's the inner cortex. And as you flow down through the inner cortex, again, the lymph is going to flow over a whole bunch of immune cells, and now it's going to be all those T cells. Where did they come from? Well, they came from the thymus. They came from the thymus and they just invaded all these lymph nodes and they just stuck in this tissue, and the lymph is going to flow past them and the T cells are going to get to work, recognizing antigens and helping the B cells out on humoral immunity, or those T cells are going to convert to cytotoxic T cells, and that will become cell-mediated immunity, and you go to work. Either way, you're just destroying foreign antigens or foreign microorganisms that are coming in to your body. Okay, and if you continue to flow down, keep going, you'll head down and you can see the medulla down here. This is all the medulla in the center. And as you come down, you kind of end these medullary sinuses as you continue through the trabecular sinus, and even further down, I'll draw an arrowhead there, down into the medullary sinuses. Now you're flowing through the medulla of the lymph node, and now you got more mature B cells, particularly plasma cells. So B cells that have matured into or differentiated into plasma cells, and they're just throwing antibodies at everything that comes through that is an antigen. And now you're just in full-on immuno mode, and your immune function is just on fire right now, just ragingly, powerfully trying to destroy things that are infecting you. Okay? And again, it's those cells just embedded in the mesh. The bottom sentence here, and you see the same thing in the spleen coming up in just a second. The leukocytes, B cells and T cells of the lymph nodes, they sit embedded within that reticular mesh, and then the lymph just flows right over them, and that lymph, the immunocells, the lymphocytes, get a chance to really go to work on the immune function against these invaders. Okay? And that's what they look like kind of on the way out the door. That's the last look at lymph node. And again, that flow through. I'll trace it one more time. Coming in here into the subcapsular sinus, picking a route into the trabecular sinus. As you do, you're coming through the outer cortex right here, where there's tons of B cells set up. Now you're moving through the inner cortex and around in here, where you got tons of T cells. I'll put a B. And by the way, these are the germinal centers. These purple blotches are germinal centers. It's just a collection of cells. In this case, out in the outer cortex, a collection of B cells. Right around the inner cortex is a collection of T cells as you move through the medullary, through the trabecular sinus. And now you get into the medullary sinus, and here's the medulla, and more B cells, plasma cells specifically inside the medulla as you flow through. And then you're all cleansed, and out you go through the efferent vessels and onto the next lymph node, and you just repeat and repeat and repeat. And then the spleen, which is oddly enough the largest singular mass of lymphatic tissue in the body. To see it green is very odd, but don't worry about it. It's sitting here in this position, and what is it doing? It's essentially a little different function. It's actually filtering your blood for pathogens and antigens. Okay. Everything we talked about so far in the lymph nodes was filtering your lymph. Now you're filtering your blood in the spleen. Okay? Same concept of filtration with lymphocytes, B cells, and T cells ready to go and fight, but now you're filtering the blood. Right? So it's located lateral to the pancreas, lateral to the stomach, superior to the kidney, on the left side of your body, tucked up high, just under the diaphragm. And it's a highly vascularized organ because now you're feeding the blood in, and you're putting a bunch of lymphocytes, B and T cells, inside of a reticular mesh, but now you're flowing blood over it, not lymph. And that's a huge difference that you really have to be able to get your head around. And how does that work? Well, the connective tissue capsule, not shown here, but you can imagine, because here's a trabecula shown really clear, diving down for flow. But you'd have the connective tissue capsule up above, connected to the trabecula. Okay, and what you're gonna, and again, all your cells are all set up around it. What really is different is the central artery shown in cross section. There's a small artery here, and the blood is flowing inside of this artery, and the blood is coming through. And you know what kind of capillaries, right? You know what kind of capillaries. I'll show you in the slide. You know what kind of capillaries are here, because lots of things are free to just sort of diffuse out of the capillaries into this parenchymal structure here called the white pulp. And it's immunotissue, it's lymphocytes, lymphatic tissue surrounding the blood vessel. And as this blood filters out of this vessel and leaks out, all that blood is being subjected to all these B and T cells that are sitting in this white pulp. And same thing's going on. Antigens are being identified, recognized, bound to. T cells are going to work to help out B cells. T cells are becoming cytotoxic and killing everything in sight. And full-on immune function going on right here. Raging immune function going on right here. Okay. The other parenchymal aspect of the spleen is the red pulp, and I'll outline a couple sections of red pulp. And what is the red pulp doing? Well, the red pulp is going to be pulling out the red blood cells, because we've talked about this back in 40B. Okay. So again, the spleen functions a little bit differently because it's filtering blood, not lymph. The white pulp is lymphatic tissue. I'll circle it one more time. Is lymphatic tissue around the central artery with B and T cells. Whoops. B and T cells going to work. Okay. B and T cells going to work, filtering blood. Okay. B and T cells filtering blood. And that's going to be the white pulp. Okay. Just follow it through the slide right here. And sinusoidal capillaries. It's all about sinusoidal capillaries, right? What type of capillary is going to be sitting here in this around this artery, and as the blood flows into the spleen, it's going to be sinusoidal capillaries, and that's going to allow all the constituents of the blood to flow out into the white pulp, so that you can get to work filtering the blood in the white pulp. And that's going to be really, really important. And within the red pulp, again, you're going to be doing more vascular work than immunological work. Okay. Within the red pulp here, as the blood flows out through the sinusoidal capillaries, you're going to be moving the red cells out into the red pulp, and you're going to be removing them, breaking them down, and taking apart broken down red blood cells, as well as broken down white blood cells, as well as platelets too. Okay, so the red pulp is more of a vascular function than the white pulp. And remember, there are no afferent lymphatic vessels in the spleen, so you do not bring lymph into the spleen. No lymph enters the spleen afferently. It filters blood, so it's not filtering lymph. You do have efferent lymphatic vessels. Why? Well, because every tissue has efferent lymphatic vessels, or lymphatic capillaries, because the blood coming through pushes out through the capillaries and enters the interstitial fluid, and you don't put as much back into the capillaries, and so you have to collect. So now we're seeing here, remember we had three functions to lymph. One was to collect extra interstitial fluid, and that's the function going on in the red pulp. We also had a small function of dietary lipids, but then we had the major function of the immune system, and that's what's going on in the white pulp. So pretty interesting little details here, okay? Spleen white pulp is immune function. Spleen red pulp is collecting and draining excess interstitial fluid and draining it back into the circulatory system. And then lymph nodules, really simple, straightforward. They're collections of, similar to lymph nodes, but they don't have the connective tissue capsule over them, so they can't be classified as organs technically. Right? No big deal. Typically scattered through connective tissue, the lamina propria underlying the epithelium and all of the mucosa. Where are you going to find those mucosal linings? Lining the GI tract, lining the urinary tract, lining the respiratory tract, the reproductive tract. And sitting in there are going to be all these MALT, mucosa-associated lymphatic tissue. Collections of exactly what you see in the lymph nodes. So collections of T cells and B cells inside a mesh. But they're not as clearly wrapped as a lymph node in an organ. They're more diffuse. The tonsils are a really good example of this and where we've seen it before. So the adenoid tonsils, the palatine tonsils, the one you get removed typically in a tonsillectomy, and the lingual tonsils. And those are just sitting there. So anything ingested, anything inhaled, right? Anything excreted through these tracts, the GI, the urinary, the reproductive, and the respiratory, is passing over this these lymph nodules, which is lymphatic tissue, and they're being filtered that way too. All right, so let's get to work. This is super simple. The lymph node is a secondary. The red marrow is a primary. The lymph nodule is just simply called lymph system tissue. Thymus is a primary, and spleen is a secondary. Couldn't be any quicker there, could we? And now let's go to the little bit longer one where we're going to tease apart a little bit more of these elements here and kind of come up with a little bit of a longer list, okay? No afferent lymphatic vessel, filters blood, not lymph. That's the spleen. Does not have a surrounding connective tissue capsule, so it's not a lymphatic organ. That's the lymph nodule. The other four above it are all organs. Site of maturation of T cells. The thymus has a surrounding connective tissue capsule. Oh, the thymus has a surrounding connective tissue capsule. The lymph nodes have surrounding connective tissue capsules, and so doesn't the spleen. Has a parenchyma and a stroma. Specifically, we talked about those with the lymph nodes and with the spleen, but the thymus has a parenchyma and stroma too because it's an organ, so I'll go 2, 3, and 4 there. 3 and 4 for sure we talked about, but I'm going to go 2 thymus because it's an organ, and most organs you can say parenchyma and stroma. You can find the parenchyma and you can find the stroma. Has trabeculae penetrating the interior of the organ. also the thymus, also the lymph nodes, also the spleen. Has a superficial cortex surrounding a deeper medulla. Okay, the thymus hit that and the lymph node hit that, but we didn't quite get that in the spleen. The spleen, we broke the parenchyma not into cortex and medulla. We broke it into white pulp and red pulp. So I'm going to go yes thymus, yes lymph node, but not spleen. H, site of differentiation of pluripotent stem cells, and that's going to be the red bone marrow. I has red and white pulp, that's the spleen. You're filtering blood, not lymph. Site of maturation of B cells happens in the red marrow. K, place from which mature T cells are sent. They're sent from the thymus to colonize the secondary organs, lymph node and spleen. Have colonies of T cells stationed to initiate immune response on filtered colonies of T cells. That's going to be the lymph nodes. Okay, on filtered lymph. Why did I not put spleen? Because it's filtered lymph here. Filtered lymph. The spleen doesn't filter lymph. It filters blood. Does have T cells stationed, but they're stationed to filter blood. M, have B cells stationed to initiate immune response. Okay, also going to be just the lymph node, not the spleen, again, because we're talking about filtering lymph, and the spleen doesn't filter lymph. And collection of lymphatic tissues, tonsils, and to filter out pathogens, those are lymphatic nodules. O has two different types of cells, dendritic and epithelial, to test out all your T cells. It's all done in the thymus. And P, the sinusoidal capillaries, to allow for massive overhauling inspection of blood elements, is for you filtering the blood. Thanks so much. I know this one ran a little bit long. I really appreciate you sticking with it. Again, they're getting us off on a good foot to understand as we move to innate immunity coming up next module, and then adaptive immunity, and really talking about how the immune system works. But here's where we set the stage. So thanks so much for staying with it. I really appreciate it, and I hope it helped.